Mitosis in the cell cycle. In this lesson, we'll be covering how we go from 1 to trillions of cells, why it happens, the cell cycle, genetic material, genes, interphase, stages of mitosis, cytokinesis, checkpoints in mitosis, and then errors in mitosis. In this slide, we can see an overview of the cell cycle, and you can see that it is actually a cycle. It's going to start here in interphase, and it's going to go around and back to interphase. It's just going to cycle over and over and over again. Zygote. All living things start out as a single cell called a zygote. A zygote is formed when a sperm and an egg cell come together. And you can see that in this diagram here. So the egg cell is the bigger one. And keep in mind, this is not to scale. So the egg cell and the sperm cell, and they're each going to have half the number of chromosomes needed to make a full person. And when they come together, we get a whole complement of chromosomes inside the zygote. And you can see I got my arrows mixed up here. This is the egg cell, and this guy here is the sperm cell. So the smaller one is the sperm cell, the larger one is the egg cell. And when they come together, they make a zygote. Now the thing about the sperm and egg cell that are kind of interesting is that this has half the number of chromosomes that you need, and the sperm has half the number of chromosomes that you need. And then a zygote has the complete 100% of the chromosomes that you need. Zygote. This single cell divides over and over again until it reaches approximately 30 trillion cells, with the average human having about 37.2 trillion. Each cell type performs a specific function within the organism, keeping it alive. So for instance, nerve cells have this particular shape, allowing it to conduct electricity from one end to the other. White blood cells come in a whole variety of different sizes and shapes because they have a variety of functions that they need to complete. And then red blood cells, you can see right here, are this donut-shaped thing that allows it to carry oxygen and carbon dioxide around the body. Why do some animals shed their skin? As they grow, some animals shed their skin to allow for further growth. Even humans do this. We just shed it in very small amounts that are hard to see. And the cell cycle allows for this to happen. If the cell cycle wasn't around, then these old dead skin cells wouldn't be able to be replaced by new healthy cells. The cell cycle. The process of zygotes turning into trillion-cell humans is all due to the cell cycle. The process of zygotes turning into trillion-cell humans is all due to the cell cycle. Cell cycle. The replication and division of the cell nucleus, where the daughter cells are exact replications of the parent cell. New cells are produced for growth and replacing damaged or old cells. Like we're talking about the skin cells. Skin cells get old, they need to be replaced. That's where the cell cycle comes in. And you can see this image right in the bottom is going to show you an overview of the cell cycle. This is our start, even though it is a cycle, so it's not really a start, but we can think of it more as a linear path. That's the start, and this is the end, where you're going to get your two new cells. The cell cycle. Within the cell cycle, there is the growth phase, division phase, otherwise known as mitosis, and cell division, which is otherwise known as cytokinesis. The growth and replication phases are completed in interphase. The division phase, mitosis, is comprised of prophase, metaphase, anaphase, and telophase. And then lastly, cytokinesis is when one cell becomes two cells. The cell cycle. So you can see an overview of what we were just talking about. We're going to start here at G1. That's going to be our growth phase. That's when the cell gets bigger. Then we're going to move to synthesis. That's where we're going to synthesize our separate set of chromosomes, our doubling of our chromosomes. Our G2, that's going to be for growth and preparation for mitosis. And then we're going to have mitosis. And then right at the end of mitosis is actually going to be our cytokinesis right there. And then it just goes back to G1. Now the cell is going to spend most of its time in interphase. That's G1, S, and G2, and only a brief amount of time in mitosis and cell division. The cell cycle. Cells don't all divide at the same time. At any given time, different cells will be in different phases of the cell cycle. And this is shown well in this diagram here. Now this is a plant cell, it's not an animal cell, but in a plant cell, you can see that you have some cells in a completely different state. For instance, here and here, totally different state, and then everything in between. Genetic material. Our complete genetic blueprint is packaged into 23 pairs, 46 chromosomes. Each chromosome pair is responsible for certain structures and functions. Each chromosome contains DNA, otherwise known as deoxyribonucleic acid, which is the specific set of instructions your body uses to make you, you. And you can see here, that's a representation of our DNA. Our DNA is found inside our cell, which contains the nucleus. And if we zoom in, we can see our chromosomes, right? And we blow that up and we unwind it, and we can come all the way down to different sections of our DNA. We're going to talk about it in a second, but certain sections are going to be specific to certain things. This might be section 1, this might be section 2, and they're going to encode or be the blueprint for different things inside that cell. Genetic material. These chromosomes are numbered 1 to 23. You get a number 1 chromosome from mom and a number 1 chromosome from dad. So for instance, that would be number 1, and that would be number 1. So this one might be mom 1, and this one might be dad 1. This would be mom 2, and this one would be dad 2, etc., all the way through. In total, you get 23 chromosomes from dad and 23 from mom. Genetic material. Pair 23, also called our sex-linked chromosomes, that's going to be here, are what determine whether you're male or female. Males are going to contain an X chromosome, so that's going to be our X, and our Y. whereas a female is going to contain 2X. So the karyotype of a female is going to look a little different. The 23 chromosome is going to have two of them that look very similar. So this would be a female, in which case it would be an X and an X. You can see that the Y is very distinct in that it's a lot smaller, and that's what makes males males. If you lack a Y, then you are a female. Genes. Chromosomes are made up of proteins and a DNA. DNA is composed of 4 compounds, A, T, G, and C, otherwise known as adenine, thymine, guanine, and cytosine. Chromosomes are divided into sections called genes. So that's what I was talking about before with those sections 1, section 2. And genes are going to be the actual instructions used to make the proteins that make us who we are. Now you can see the way they bond in this picture on the right-hand corner here. You're going to have our different, so that's our adenine, for instance, it's going to bond with our thymine. And you can see how it bonds. It bonds right there and it bonds right there. If you think of what the double side of the helix is going to be for our DNA, if we look back here at this picture, you can see how it's got a double helix, we call it. So it's got, it's one side and it's the other side, and they're going to intertwine together. We would show that with this. So this would be the one side, and that would be the other side. Genes. Genes are short segments of DNA which tell the cell to make certain proteins. The proteins are what gives us our individual characteristics. Now, each person's genes are different except what? Except identical twins. Identical twins have the exact same genetic makeup, and that's why they're so similar looking. Essentially, at the cellular level, they are copies of each other. And that's what makes them look so similar. Genetic features. So some genetic features that everyone thinks about are things like eye color. We have brown, blue, green, all different shades of eye color. Other things like earlobes, maybe you're less conscious of this, but if you look at the side of someone's head, at their profile, sometimes earlobes are actually attached to the head, like we have here, and sometimes earlobes flap, like we have there, and that's a genetic trait. Another genetic trait is actually being able to roll your tongue. So some people can roll their tongue because they have a certain muscle that allows them to do it, while others lack that muscle and therefore can't roll their tongue. Try it, and then go home and see if your family members can do it as well, or can't do it just like you. You'll find some similarities within the families. Human-animal gene similarities. Now, genome-wide variation from one human being to another can be as much as 0.5%. That's a 99.5% similarity. That's a huge, huge similarity between one person to another, considering how different we all are. Chimpanzees are 96 to 98% similar to humans. Cats are 90% similar to humans, 82% with dogs, 80% with cows, 79% with chimpanzees, 69% with rats, and 67% with mice. So there's a lot of similarities across these different species. Cows are even 80% genetically similar to humans. 75% of mouse genes are equivalent in humans, and that's very important because this allows us to test a lot of our medications on mice because mice have a lot of the same genetic characteristics as we do. The fruit fly, or Drosophila, shares about 60% of its DNA with humans. So for a similar reason, we're able to do a lot of tests on fruit flies, which are plentiful and very easy to replicate. And about 60% of chicken genes correspond to a similar human gene. So we're not as different from each other as we might have thought. Interphase. Interphase is the time between cell divisions. During this time, the cell undergoes DNA replication and growth. The cell spends most of its time in interphase. Now we're going to start from the bottom of the screen here, and we're going to kind of repeat what we were talking about earlier. So G1 phase, that's when the cells carry out the metabolic activities to prepare for the S phase. Then we have S phase, which is otherwise known as our synthesis phase, where we're going to synthesize or replicate our DNA. And then we have our G2 phase, where organelles and molecules required for cell division are produced. And this is where our cell is fully preparing for mitosis. So in interphase, chromosomes are condensing. And you can see this going from left to right. On the left, that's our interphase nucleus. Pretty bland, you can't really see differences between the different things. In the second picture, our chromosomes are condensing. And then in our right picture, our chromosomes are fully condensed. And you can see the difference between picture 1 and picture 3. The chromosomes are much more noticeable, and they're much more distinct from one another. So here's our basic overview of mitosis. We're going from one cell to two cells through replicating our chromosomes and then splitting them in half. Mitosis and cytokinesis. This is the process whereby a cell will divide to produce two new identical cells. It allows organisms to grow and replace old, damaged, or dead cells. It occurs in all body cells. Mitosis has 4 main phases: prophase, metaphase, anaphase, and telophase. And cytokinesis is the actual splitting of the cells, like we have here. So first up, prophase. Prophase is the first and longest phase of mitosis. Strands of DNA condense and thicken to form visible, duplicated chromosomes. These are sister chromatids, and that's what we have right there. Sister chromatids are held together by centromeres, and this is where they cross. So if we take a look at this guy right there, the centromere isn't shown very well, but it would look something like that. It's the part right in the middle, right where the chromosomes cross each other, and that X is formed. The nuclear membrane is also breaking down at this point, and that's shown by this dotted line here around the membrane. Prophase. The centrioles, that's the orange things here, are going to move to opposite poles of the cell. the spindle fibers begin to form from the centrioles. And the spindle fibers are these blue things right there. You can see them over there as well. And here's another picture of prophase. Metaphase. Metaphase is the second phase of mitosis. Spindle fibers connect the centromere of each chromosome to the poles of the spindles. And you can see the spindle fibers here connecting right to the centromeres. And spindle fibers help chromosomes line up across the equator. So the spindle fibers are going to push and pull these chromosomes at the centromere until they're lined up right in the center, right along the equator, like we have pointed out right there. And that's a picture of metaphase. You can see the chromosomes. It's a little harder to tell, but you can see how they're lining up right near the middle. Anaphase. Anaphase is the third phase of mitosis. Centromeres are split apart. Each chromatid pair splits and are now called daughter chromosomes. Spindle fibers shorten and thicken, pulling one chromatid. So these are the chromatids right there, and there, and there. Pulling one chromatid, so a chromosome, from each split pair to the opposite poles. Here's a picture of anaphase. Again, you can see them being pulled apart. I find this picture on the left a little bit easier to see. And you can see all our chromosomes here, all these green guys here, they're being pulled to opposite ends of the cell. The picture on the right is going to be more of early anaphase, so it looks a little bit similar to metaphase. Telophase. Telophase is the fourth and final phase of mitosis. During this time, chromosomes gather at the opposite ends of the cell. So you can see here, we have one end here and one end here, and you can see the chromosomes gathering in these spots. At this time, also, the chromosomes begin to unwind and are less visible. The nuclear membrane begins to reform, and we're showing this with the dotted line that you can see right there. And spindle fibers begin to break down, and we can see that by the shortening of the blue lines, which are spindle fibers. Cytokinesis in animals. After mitosis occurs, the cytoplasm separates and two identical daughter cells form, and that's what we're seeing here. This is going to be daughter cell 1 and daughter cell 2. Cytokinesis in plants. In plants, a structure known as the cell plate forms midway between the divided nuclei, and we're seeing that right there and right there. The cell plate gradually develops into a separating membrane, and a cell wall begins to appear. So eventually you get this hard cell wall dividing up the two new cells. Now there are certain checkpoints in the cell cycle. Now a cell will not divide if signals from the surrounding cells tell the cell not to divide, if there's not enough nutrients to provide for cell growth, if the DNA within the nucleus has not been replicated, and if the DNA is damaged. Errors in mitosis. Substances such as tobacco, asbestos is used in building materials, radiation, and viruses can cause mutations. Mutations alter the structure of our DNA. It's going to change those A, G, C, and T's into something else. So it might change an A into a T, for instance. And if you do that, then you're going to get a completely different strand that'll code for a different protein, which will result in an error. When these cells divide, the mutation is passed only to the daughter cells, but it can spread throughout the body. One result of a mutation can cause cells to divide uncontrollably, leading to cancer. So, for example, cigarette smoke can alter the chromosomes in the lungs, causing these cells to undergo mitosis much faster than normal, as we're seeing right there. Now, these cells are going to have a different structure and function, and can lead to issues with breathing, and can further develop into other problems down the line. Essentially, this is lung cancer, because it's cancer inside the lungs. Retinoblastoma is another kind of cancer. It's a cancer of the retina. The retina is located on the back of the eye, and if you get a cancer in the back of the eye, it can be retinoblastoma, which is a rare cancer caused by mutation to certain genes in the retina, which are carried on through mitosis. And that concludes our lesson on mitosis.